Okay, perfect. All right, I guess I get started. Well, thank you, everybody. It's an honor to be here presenting to you today. Okay, I'll talk to the microphone. There you go. Thanks, Mike. Thank you very much for inviting us to talk here. It's an honor to be here to tell you about my research. Today, I want to tell you about some of my work trying to understand the processes of structured communities at multiple spatial scales. And today, I'll give you an overview of some of my research, both throughout my PhD and my postdoc. So I've been trained as a community ecologist, and most of my time I've spent thinking about, I should have started then. I've spent most of my time thinking about how species interact with one another, both in food webs, but more recently as well in some mutualistic interactions. And throughout my PhD, I kind of developed this love for statistics and data science. And I've brought it to as much of my work as I can. And so some of my work includes, doing large-scale biodiversity analysis, data management and cleaning pipelines, different types of ecological simulations, as well as the development of statistical methods. And so these kind of were the two aspects that I try to leverage the most during my PhD. And throughout my postdoc, I actually wanted to focus this into and apply it to a conservation aspect. And particularly, I've been thinking a lot about the problem of insect and pollinator declines. And so with this intro, today I'm going to tell you about some of my work linking experiments and theory of bromeliad food webs, as well as how I've been analyzing occurrence data to estimate trends of biodiversity through time. Okay, so first, linking experiments and theory in bromeliad food webs. So bromeliads, in case you don't know, they're an amazing set of plants. They're a great study system because they provide habitat for a bunch of macroinvertebrates. So bromeliads have interlocking leaves that kind of collect water, and within the water, you get communities of aquatic invertebrates. And these aquatic invertebrates form a food web where we have a bunch of different types of prey, chironomid larvae, culex larvae, tipulate larvae, and a very voracious predator that eats everything that you put in front of it. So I went down to coastal Brazil at the Jurubatiba National Park, and I collected a bunch of these individuals to do some feeding experiments. And what I was interested in was understanding how differences in body size of prey affect the local food web stability. One of the cool things about these food webs is that it has a very large gradient in body size, where some of the prey are very small and some of the prey are quite large. And I wanted to understand kind of what's the effect of these body size gradient. And so I did this feeding trials with this generalist predator with multiple prey species. So you can see here the damselfly with one of its prey, and there it is enjoying it like a little spaghetti. And so for each of the species within the food web, I offered all of these species to different individual predators of the same species. And so on the x-axis, we have the number of prey. Oh, here it is. There you go. The number of prey offered. And on the y-axis, we have the number of prey consumed. And as you can see, the predator saturates, right? So this is a type 2 functional response where there's a point where the predator says, okay, this is enough. I can't eat just as quickly and everything that you provide to me. But the cool thing is that the saturation point varies as a function of body size, where the predator saturates faster the larger the prey. This experiment allowed me to parameterize a functional type 2 response, where I fit the attack rate and the handling time as a function of body size. So the first result of this experiment was that the attack rate decreased with body mass, while the handling time increased with body mass. Now, with these two relationships, I was able to build a multi-species predator-prey model, where the prey abundance was determined by the prey abundance in the previous time step, plus new prey added via population growth based on the prey body mass, minus competition based on body mass. So 2 species that were closer together in body mass competed more strongly than 2 species that were further apart, minus predator consumption based on the prey body mass, and that was the parameterized functional response. Similarly, for the predator abundance was determined by the predator abundance in the previous time step, plus new predators added via the consumption of all of its prey, minus predator mortality. And as you may notice, all of the parameters in this model are dependent on the prey body mass. That's really the only toggle. So what happens when we look at this model? So the first thing I want to look at is when we have one prey and one predator. Now, it's only at this intermediate body sizes that both the predator and the prey persist. When the prey is very small, the attack rate's very large. So the predator consumes its prey very quickly, drives it extinct, goes self-extinct. When the prey is very large, the handling time is very long. So the predator cannot obtain enough energy through consumption, it goes extinct, and the prey is allowed to persist. So it's only at this intermediate body sizes that both the prey and the predator persist through time. But as a community ecologist, I wanted to know what happens with more species as we increase diversity. And previous studies had shown that the stability of a food web depends on the consumer-resource ratio as well as the diversity of the food web. But I wanted to break down some of the consumer-resource ratios as well as include this nonlinearity in attack rate and handling time. Now, even though the only parameters in my model were the body size of the prey, as I increase prey diversity, the number of prey species, the combinations in parameter space became very, very, very large. Like this is in log scale. So the number of combinations became computationally unfeasible to run the model. So in order to overcome this problem, I use an MCMC strategy where basically I was doing an MCMC with simulation space, where I was able to reduce parameter space by searching only in the interesting areas. For example, persistence or not persistence of the predator. And this allowed me to reduce parameter space from 10 to the 20 combinations of parameters to 20 to the 4. So it became a much more manageable problem. Okay, so let's start increasing diversity. So here in the x-axis, we have the body mass of prey number 1. On the y-axis, we have the body mass of prey number 2. The areas in yellow are the areas where the predator and all the species persist, and in purple is where it doesn't persist. Now you can see that the persistence of the predator is constrained at this intermediate prey body sizes. But one of the interesting things is that it's only this prey number 1 that actually constrains the persistence of the predator. As long as prey number 2 is larger, it doesn't really matter what body size it is. So that's cool. And as we continue to increase diversity, again, the predator's persistence was only constrained by this smallest, like the body size of the smallest prey. But the interesting thing is that the range of potential body sizes that this medium-sized species could have increased and then decreased with diversity, right? So here, the possible range of body sizes actually increased and then decreased. And this is partly due to these nonlinear relationships between attack rate, handling time, and body mass. And so overall, I found that prey diversity had this unimodal relationship with predator persistence, where the predator was most likely to persist at this intermediate number of prey species. Okay, so this was just kind of a fast showcase of one of the projects that I've done, but also throughout my PhD, I was interested not just in thinking about the food webs at a local scale, but understanding what's happening at larger spatial scales. So some of my other work includes estimating dispersal rates for a couple of species in this food web using genomic tools, and then incorporating both these attack rates and dispersal rates in a meta-community model to try to understand the persistence of these food web, both at local and at regional scales. And the bromeliad system was really cool because it allowed me to think about meta-communities more broadly. And so some of my work also includes thinking about other kind of conceptual theory that we need in meta-community ecology in order to understand how food webs and competitive interactions persist at local and regional scales. So now I want to spend the rest of the time telling you some of my most recent work, thinking about how we can analyze occurrence data to estimate trends of biodiversity through time. And we live in a really interesting time during biodiversity because the amount of data that we have for a lot of species is increasing very rapidly. Museum collections are being digitized very quickly. And community science apps are allowing us to collect occurrence data for biodiversity at unprecedented speed. And so apps like iNaturalist, Bumblebee Watch, eButterfly, eBird, all of these different tools are allowing us to collect biodiversity data. And I pulled out this plot a while ago. And, you know, even a few months ago, the number of occurrences in GBIF alone was over 120 million. And so the number of records that we have for species is incredibly large. Now, one of the problems, though, is that the data is quite biased. So this is occurrence data for bees in North America. And as you can see, the number of occurrences rapidly increases through time, both data that has been digitized by museums as well as observations from iNaturalist. And if we analyze this data naively, we may think, well, you know, bees are doing great. They've been increasing through time. And, okay, similarly, the data is quite biased spatially. And again, this is the same records, but plotted over space. And if we analyze this data naively, we may think that US cities are some of the most biodiversity hotspots for, for bees, right? And so clearly, you know, we need to take into account, like, while these data seems very powerful, there's a vast amount of data, there's a lot of things that we need to consider if we want to analyze this data. And occupancy models provide us one potential route to analyze this data. So first, I want to walk you through kind of understanding what an occupancy model does, and then I want to tell you what are some of the things that you have to consider when applying occupancy models to occurrence data. So imagine we set up 4 different sites, and we visited those sites in 3 different time intervals. Now, when we're dealing with the structured data, the first thing we do is we record both the presences and the absences, right? And so we see that every time we visited site number 1, we observed our bee in every single time interval. When we visit site 2 and 3, there's a couple of times we did not observe this bee, right? But we have a pretty good record of detecting this bee when we go there. So these 2 cases are probably missed detections. On the other hand, in this site, we never observed this site, even though we went to this site 3 times. But because we have a pretty good record of detecting species when we visit sites, this is probably a true absence. Now imagine a more tricky scenario where our ability to detect species is very poor, and only 1/3 of the times that we go to a site, we actually observe the species. Now, if in this particular case, in site number 4, if we don't observe it, it's harder to tease apart whether this is just a misdetection or indeed the species is absent in here. And so occupancy models really help us disentangle these 2 types of zeros, where we have a misdetection versus a true absence. That's the role of occupancy models. And so today I want to kind of summarize 2 of recent studies that I've done where we try to also identify some of the pitfalls that, or some of the complications that we have to think about when we're applying these occupancy models to occurrence data. So the first one is that when we're talking about occurrence data, we only have the presence of the data, right? We only have presence-only data. So we may be able to bin this data in space, bin this data through time, right? We bin it into multiple sites, multiple time intervals, but at the end of the day, we only have the presences. Now, a naive analysis might just say, oh, well, these are zeros, right? We fill in the site time interval combinations with zeros. But the problem is that some of these sites may not have been visited in those particular time intervals, or they were visited but not in the database, right? A lot of the data is still being digitized. Or it could be a case where that site was visited and no species was detected. So we have to think about how we can perhaps disentangle some of these things. Another thing that we have to think about is collector behavior. So imagine we have a community of a bunch of different types of beetles, and we have two types of collectors. We have somebody who likes to target individual species. So this particular person only cares about orange beetles, only visits sites that have orange beetles, right? And so only visits and cares about these orange beetles, doesn't even visit the second community. But we also have other types of samplers, which are community type samplers. They visit every single place that has a beetle. They collect every single type of beetles that they observe. And so if we consider these two types of behavior, the resulting collections may look something like this, where the person that only cares about orange beetles only records the orange beetles. The problem is that the data that we get looks like this, where we don't actually know reasonably well what the underlying community is, nor do we know who, what was the purpose of the original collection, right? In a lot of these databases, we don't, besides from recording the name of the person who collected the data, there's no other metadata that tells us what this person wanted to do. So we almost have to try to work around those two types of collectors and or try to guess what people wanted to do in the first place. And so in order to figure out how to overcome these two different challenges, we simulated data that was like historical records. So here we have a bunch of different sites. We simulated multiple time intervals within an era, where we were able to estimate occupancy within an era and then also changes in occupancy between eras. So you can think about changes in a species distribution through time. We also simulated ranges for each of the species. And then we also simulated a community. And so in the simulations, we simulated a case where a particular species of interest was detected. We also simulated cases where other species in the community were detected, but not the particular species of interest. We simulated cases where a visit occurred and nothing was detected. And because we were in simulation land, we could actually do this. We knew when a visit happened and nothing was detected. And we also simulated cases where no visit occurred. And part of the goal of our study was to try to figure out, okay, well, what modeling choices can we do to actually infer the correct trends in change in occupancy through time? And so in the naive way, we may include every single site and every single time interval for every single species, right? So these black dots just represent every single square that was included in the model. But the problem is that if we model species outside the ranges, that introduces zeros that couldn't have been a misdetection or a true absence. It makes sense. But also, modeling species in sites that were not visited also introduces zeros that could not have been a misdetection, even if it's within a species range, right? So in that particular time interval, a visit actually never happened. And so these two things, modeling species outside the range introduces zeros that couldn't have been misdetections, modeling species that were not, modeling sites that were not visited also introduces zeros that could not have been a misdetection. Both of these extra types of zeros that are not misdetections nor true absences bias our detection probability, which then biases our estimates of occupancy. And indeed, what we find is really troubling. If you do this, if you do the naive scenario, where you include every single site in every single time interval, you confound changes in detection and changes in visit history with changes in occupancy. So here, the dashed line, it's the true change in occupancy. We simulated no change in occupancy through time. Instead, we also simulated changes in detection probability, so species are easier to find through time. And we also simulated changes in visits, so more visits occurred or less visits occurred through time. And what we find is that only when these two kind of detection probability and visit probability are at 0, so they don't change, we're able to estimate changes in occupancy. But the moment that any of these changes, we assume that changes in detection are actually changes in occupancy. And we know that this occurrence data, I showed you the graph through time, the number of records and potentially the number of visits has been changing through time, right? And so if we do this naive approach with occurrence data, we may be getting into a lot of trouble. Now, another type of approach, we may be able to restrict species to their ranges or sites that could be suitable. And another thing we can do is infer absences based on other species presences, right? So we take all the species, for example, for bumblebees, we take all the species that are bumblebees, and every time we detected a species, we assume an absence for every other species. And the good thing about doing this, even though we miss the cases where a visit occurred and nothing was detected, is that at least we have positive evidence that a visit actually occurred. And indeed, when we do this approach, we find that we're able to recover changes in occupancy, and so we're able to recover changes in occupancy when occupancy doesn't change, when occupancy changes, when detection probability changes, right? So you can see that all of these kind of confidence intervals overlap the gray line. So that's good news. And so inferring non-detections using other species and restricting species to the ranges results in accurate estimates of probability of occupancy. That's awesome. But there's one problem. This type of approach assumes that information about a species' presence tells us something about other species. And this is only the case when species were looked at as communities, right? And so this assumes that everybody is a community collector and that every single visit was a community collection because we're using other species' information to infer their absences. But we know that targeted species sampling occurs. Let's say you're a taxonomist and you want to do a taxonomic revision in a particular group. You only target that particular group that you're trying to find. Let's say that you're a community scientist and you only take pictures of the shiny red things you observed in your garden, right? So we know that targeted collections actually occur. And so we wanted to know how robust are our inferences to these type of behavior. And so we model this type of behavior also in our simulations. Now, here on the x-axis, we have the probability that these are that a visit is a community collection. So when this probability is number 1, every single collection is a community collection, right? And so this basically is the case where we can infer absences based on other species presences. And indeed, so over here on the y-axis, we have the error of estimated occupancy. We find that we're able to estimate occupancy with very little error. On the other end, when this community visits is 0, the probability of community visits, so every single visit is a targeted visit, we see that our estimates of occupancy have a lot of error, right? So these make sense. If none of them are community visits, we don't have any information about absences. If everything is a community visit, we have lots of information about absences. The good news, and I think this is like the nice moral of the story, is that even if you have 50% of community visits, even if 50% are targeted samplings, you can still do this approach. You can still do this. And so that's awesome. It's not all, bad news. And so we took these 2 studies and we took all this approach to actually figure out what's been happening with bumblebees in North America. And so this is a project led by Hannah Jackson, and we were trying to both estimate changes of occupancy through time for 46 species of bumblebees in North America. And we wanted to know whether we could attribute some of these changes in occupancy using climate, like temperature, precipitation, changes in land use. And because I've been telling you a lot of results about many different things, I'm not going to go into the nitty-gritty details of the story, but I just wanted to highlight the first result is that there's lots of species-specific responses, right? And so you may want to ask, how are bumblebees doing? Well, that's a really hard question to answer. And in fact, we find that, you know, a bunch of species are increasing, a bunch of species are decreasing, and the vast majority are stable, at least in occupancy. And so we find, for example, Bombus impatiens, that it's increasing through time, Bombus affinis, which is decreasing through time. The only takeaway here is the variety of the colors, because I know you can't really see the numbers. But even their responses to temperature are also very varied. And so some species increase their occupancy with temperature, some of them decrease their occupancy with temperature. Now, the other thing that we found in this study is that generally the changes of temperature in the past century have negatively affected the majority of the species, even if their occupancy has remained stable or increasing through time. So that's the sad news. And the next step for these type of projects is to try to apply this to about 3,000 species of native bees in the US. And so this is a project that I've been doing with iDigBees, actually trying to infer changes in occupancy, changes in native bees across the continental US, which we can only do with these type of approaches because we don't have very long time series across the entirety of the landscape. And I think I have a little bit of time for questions. And so with that, I want to thank you very much for listening, and I'm happy to take questions. Yeah. Problematic the sampling problem is? 100%, yes. So you would expect our ability to detect species to be lower, right? But one of the good news is that even if our overall detection probability is lower, occupancy models can account for a lower probability of detection or changing detection probability both across space and time. The other thing is that we bend the data both in space and time, right? And so one of the things we can do is say, does it make sense to have a site be 100 by 100 meters or 100 by 100 kilometers, right? And so at a scale of, let's say, 10 kilometers or 20 kilometers, we may not expect to have the same problems with mobility that we would with a site that's 100 meters by 100 meters, right? And so as long as we choose kind of reasonable spatial and temporal scales for the life history of the species, that helps us with some of these problems. Yep. Yeah, so eBird does an excellent job with this, where you have a checklist, incomplete checklist, or just like random observations. And they actually label them as like this visit was a checklist, like a full checklist where they actually look for everything. iNaturalist depends on the project, maybe a little bit more disorganized because in iNaturalist you can create multiple types of projects. And Europe is a lot better at this than, like some of the community science approaches in Europe are actually a lot more structured. And so that data is a lot easier to analyze. But for bees, in North America, less so. But I think there is like a changing, like people are more aware of these problems. Yes. Okay, that's my cue. Thank you so much.